Hi, everybody. I'm Tom Roberts from Oxford. I'm a molecular biologist. I've been working on DMD for many years now. And today, what I'd like to do is give some basic biochemistry introduction. This is primarily aimed at the parents in the audience, and hopefully that will be useful to some of you. And then I'm going to outline the likely next developments in the exon skipping space. And then I'd like to talk about some new data from my group, which concerns a comparison of exon skipping with CRISPR-Cas9-mediated gene correction. So a key concept in molecular biology is the flow of information from DNA to RNA to protein, which is illustrated here. And you can think of this an analogous situation is a recipe, with the genome being a recipe book, and then we make a temporary photocopy of a recipe, and then that is translated into a meal. So in the context of DMD, we have the gene which encodes the dystrophin protein. This is transcribed into a dystrophin mRNA, and then that encodes for the dystrophin protein. And in DMD patients, what we see is that there's a mutant copy of this gene that results in a mutant mRNA, and as a result, dystrophin protein is not produced. So the other biochemistry concept, which is really important to understand exon skipping, is the idea that genes are actually discontinuous. They consist of coding regions called exons. They're interspersed with non-coding regions called introns, which you can consider these to be a little bit like punctuation. And through the process of RNA splicing, these exons, the coding regions, are stitched together and the introns are removed. So if we consider the dystrophin gene, it's actually an unusually large gene. It takes up a very large chunk of genomic real estate, if you like. It has 79 exons and 78 introns, and it's depicted here. So each one of these vertical lines is one of those exons. And this encodes dystrophin, and you can think of dystrophin as being a little bit like a shock absorber. It protects skeletal muscle fibers from contractile damage. And you can see here that we have, I'm not sure if the laser's working, no, it's not, but we have one part of the dystrophin protein which is connecting to the inside of the cell, and one that is connected indirectly to the outside of the cell. And then we have these internal regions which are a little bit like the coils of a spring. And the reason why I use this image is that you can imagine that a spring would work still as a spring with fewer turns of those coils, but perhaps not as good as a spring. And that's essentially what we're trying to achieve with exon skipping. So there are a number of dystrophin restoration approaches. This was mentioned a little bit earlier. So exon skipping I'm going to talk about today. Gene replacement, there's a session tomorrow. Stop code and read through has been mentioned. Gene editing and cell therapy. And today I'm going to concentrate on these two. So with exon skipping, here is an example of a chunk of the dystrophin gene, a cartoon schematic, where we're seeing exons 45 to 53. This is a healthy individual. And you can see that with each exon, there's little chevrons, little arrows, and these connect together like a puzzle, and that's required for the code of the message to be correctly translated. So after splicing, the introns are removed, and these exons are stitched together. We get a functional dystrophin protein. Now, in the case of DMD, I'm showing here an example where exon 50 is deleted, which is a relatively common DMD-causing mutation. Now, after splicing, you can see that these chevrons don't match, and the code is now disrupted, and that results in failure to produce a correct dystrophin protein. So with exon skipping, what we aim to do is to make an internally deleted and partially truncated protein by skipping an exon, in this case, exon 51. And now you can see that exon 49 and 52 can splice onto each other, and we get this in-frame dystrophin transcript, resulting in the formation of a functional protein, but with fewer turns of those internal loops of the coil. The biochemical, there's an analogy for the spectrin-like repeat domains of the protein. So here you can see the outcomes: healthy at the top, DMD non-functional, and exon skipping partially functional, internally truncated. So there are a number of FDA-approved therapies for DMD, and these are shown here. They target various exons, 51, 53, and 45 from two pharma companies. And these are all consisting of the same type of chemistry, which we call PMO, which stands for phosphorodiamidate morpholino oligonucleotide. Now, the key point is that the effectiveness of these drugs is very low, on the order of about 1% of dystrophin, of healthy levels of dystrophin restored after treatment. But we do know that these drugs are very safe, and they can be dosed at quite astonishingly high doses in animal models. Importantly, none of these drugs are approved for use of patients in the UK and Europe, and their approval in the US was actually a very controversial decision, primarily as a result of the very low efficacy and the low numbers of patients in the trials that supporting the initial, especially in the case of eteplirsen, the first drug. And a key barrier to success is drug delivery. Can we get more of these compounds into the muscles of DMD patients, and especially into the heart? So now I want to quickly outline sort of what kind of developments we're expecting in the next few years. So the drugs which I mentioned on the previous slide, which are shown here highlighted in bold, target these exons 51, 45, and 53. And you can see that these are the biggest single chunk of exons that could be used to treat DMD patients. But skipping of those exons could be used to treat DMD patients. So there are lots of other exons that are skippable that could also be of benefit, but the proportion of patients to which this applies is kind of getting less and less. But this is the kind of summary of the pipelines for the various companies from what's publicly available. So the companies are listed on the left, and you can see I've put green boxes for the exons that they're currently developing drugs for. The ones that are approved are shown with text. So the key message here is that exons, or patients with skippable 44, 50, 52, and 55 are likely going to be the next target patient population that could be treated with exon skipping drugs. So I want to switch gears and talk about delivery for a moment. So the leading strategy in the DMD space for improving the activity and the delivery of these oligonucleotides is what we can sort of summarize as bioconjugation, which just means attaching some compound to the oligo, which is going to help it get into the muscles of these patients. And of this strategy, the leading approach is peptide conjugation, and the technology is referred to as PPMO, or peptide PMO. So the PMO drugs that I spoke about on one of the previous slides, imagine that we just add a extra compound onto one of those oligos, which now improves its delivery. And that's exactly the case with the first one that I've listed here, which is SRP-5051, which is the Sarepta drug. So this is the exon 51 skipping drug eteplirsen, which is approved in the US, with a short peptide attached onto it, which helps get those oligos into patient muscles. But there are a couple of other companies in this space. PepGen, which was founded by my mentor, Matthew Wood, is in phase 1 currently, and Entrada has a similar technology. Key thing about PPMO is that they are much, much more potent than unconjugated naked PMOs, and they also show activity in the heart, which I think everyone in this room knows is really an important challenge for treating DMD. So there are a couple of other bioconjugation strategies. One is antibody conjugation. This is being pioneered by Avidity Bioscience in the US, where they've attached a transferrin receptor antibody to these oligonucleotides. And a similar variant of this from Dyne Therapeutics uses, again, a transferrin receptor targeting its antibody-related approach using a Fab fragment. So it's a smaller chunk of an antibody. These are, I think, Dyne is at early clinical trials, I think, in the last, very recently. So one slide of data, this is taken from the Sarepta website, just to illustrate this point that PPMOs really are more effective than PMOs. So you can see on the left with the orange bars, This is 30 mg per kg treatment with eteplirsen, and then the dystrophin Western blot, and so measuring dystrophin expression 24 weeks later. And you can see that the levels are less than 1%. That's on the far left. And then on the far right, we can see DMD patients treated with the peptide PMO conjugated version of eteplirsen. So it's the same PMO, but with a different delivery technology attached to it. And you can see that there's about an 8-fold increase in dystrophin production, and that's after only 12 weeks. So it's not quite a direct comparison. So this is very encouraging, and based on a lot of work that we've done in our lab using PMO technology, this is very much what we see as well. So we've talked about this idea that, it's assumed that therapies that can restore dystrophin in patients' muscles will correct the disease, and there was, some interesting discussion earlier about maybe there's some caveats to that point. But this is the fundamental principle underlying the kind of therapies we're talking about here. And key determinants of therapeutic success could be considered to be the amount of dystrophin that we put back, the quality of that dystrophin, and bear in mind we're talking about internally truncated versions of dystrophin in the case of exon skipping, and also with gene therapy, which is what you'll hear about more tomorrow. But critically, the correct localization of dystrophin at the membrane of the myofibers of DMD patients. And this is what I'd like to talk about today, and I think this is a point that has been somewhat neglected. So a few years ago, we published a paper that basically argued that a uniform pattern of dystrophin localization at the membrane was important for therapeutic success. And we did this using a genetic mouse model, which is called the MDX-XIS mouse, which is shown in these bottom panels. And we observed that there was a patchy pattern of dystrophin in these mice. And the reason for that is that they express dystrophin at a variable level because of skewed X chromosome inactivation. But that's a kind of technical detail. But when we look at these mice here, you can see that they still have regenerating fibers. So they have quite a substantial amount of dystrophin produced in a patchy manner, but it hasn't stabilized the turnover of muscle that we observe in DMD patients. And by contrast, the PPMO-treated mice, which express similar levels of dystrophin, there's no sign of regeneration. The muscle has been completely stabilized. So the regeneration here is these red-stained cells, which we're staining for embryonic myosin heavy chain. So what I was really interested in was whether we could look at the various kinds of dystrophin restoration therapies and ask the question, do these restore dystrophin in different distribution patterns? And we were really interested in comparing exon skipping with CRISPR approaches. So I'm just going to quickly run through these studies now. Oh, and to contrast the different approaches, exon skipping would involve reframing the transcript at the RNA level, which then results in re-expression of dystrophin. And by contrast, CRISPR involves correcting at the DNA level the dystrophin gene, and then that results in a corrected mRNA and re-expression of a protein. So they're somewhat similar in that in both cases we're trying to remove one exon from the genome or the transcriptome, but this diagram illustrates that we're attacking the problem at different stages of gene expression. So for exon skipping, we designed a single ascending dose study in the MDX mouse using one of our favorite peptide PMO conjugates, which is shown diagrammatically on the right. And we observed dose-dependent dystrophin restoration, fairly substantial levels of dystrophin, median level of about 50%. So this is much, much higher than what you would see with a naked PMO technology. And we found that this also resulted in a dose-dependent restoration of circulating biomarkers in these patients, which was also very encouraging. But importantly, when we looked at the immunofluorescence staining to see where the dystrophin was in these fibers, we see a relatively uniform pattern of expression within fibers. There is some in the 3 mg per kg dose, which is in the middle of this panel. You can see there are some positive fibers in a sea of negative fibers. But when we look in the longitudinal section, so imagine looking in this orientation, the dystrophin expression is largely uniform. So we wanted to look at gene editing for comparison. And gene editing is basically a technology that has the potential to achieve permanent exon skipping. So instead of altering the splicing pattern to remove that exon from the RNA, we can go into the DNA and cut that exon out permanently. But there are lots of important caveats and limitations of this approach, which perhaps we can get into a discussion of that later. But I want to talk about one of them today. So here's the approach. We're trying to remove exon 23 in the MDX mouse. Oh, sorry, this is actually in the DKO mouse. This is a utrophin-dystrophin double knockout. It's a very severely affected mouse model. It has the same mutation as the MDX mouse, but those mice die typically within 8 to 10 weeks of age. So we use two guides, which will make DNA cuts on either side of the exon, and then the cell's DNA repair machinery will stitch those together, and then as a result, all the transcripts which are made from that gene will be skipped. So you'll have forever exon skipping is the concept. And to achieve this, we use a pair of viral vectors. It's a type of virus called AAV. And we inject the animals basically as early as we could and sacrifice them at the humane endpoint. So we were able to see sporadic evidence of editing at the DNA level in multiple tissues. This is shown in panel A, and we confirmed this by direct sequencing. And then we were also able to detect skipping at the RNA level and dystrophin restoration at the protein level in all tissues tested. But the levels of dystrophin expression are very low, on the order of 1 to 5% of healthy. So much, much less than what is observed with our relatively highly optimized PPMO approach. Now, this amount of dystrophin that we were able to put back was not sufficient to extend lifespan in this mouse model. And that's one of the key advantages of using this mouse. Because it succumbs to the disease so early, a therapy which is effective can be measured in terms of lifespan extension. And we didn't observe any lifespan extension in these animals, suggesting that we hadn't put back enough dystrophin or there was some other problem which was preventing that. And when you look at the immunofluorescence staining for dystrophin in these animals, we see a number of interesting features. So the CRISPR-treated animals are on the bottom panel. I'm sorry, I'm standing slightly in the way. But what we noticed was that there was incomplete dystrophin staining around the circumference of many of these myofibers in the treated animals. And a similar observation has been made by Francesco Montoni's group in human DMD patients and also patients with mild Becker's and intermediate muscular dystrophy. And they reported that there was better functional performance in the patients that had a higher degree of myofiber circumference coverage of dystrophin. So when we look at longitudinal sections from these animals, and these are the bottom two panels in this plot, you can really see that there's patchy dystrophin in these fibers. You have regions of dystrophin-positive membrane with dystrophin-negative regions directly adjacent. And the reason for this is that by using a CRISPR approach, we've essentially made what we call heterokaryons. And these are cells that contain different types of nuclei. And I propose that there are three kinds of nuclei in these treated myofibers. We have corrected myonuclei, where the CRISPR machinery made cuts to the DNA, and this was repaired in a productive way, meaning that those myonuclei can now make dystrophin. We have uncorrected myonuclei, where the machinery, the therapeutic machinery just never got to those nuclei, and the DNA is unchanged. But we also have corrupted myonuclei, and these are nuclei where the CRISPR machinery made cuts and the DNA was repaired, but the resulting repair did not result in the production of a productive dystrophin gene. I think this is a really, really key point that has been perhaps a little bit ignored. And I propose that while these non-productive repair events occur, you will always get these mixed fibers with myonuclei that both express and don't express dystrophin, leading to a patchy expression. And we performed long-read sequencing of the edit sites to try and detect what was going on. And we detected all kinds of things that would be undesirable. So a very common occurrence was asymmetrically indel formation. So where the CRISPR machinery cut at one site but not at the other. And then as a result, the target site was disrupted, so it could not be recut again. But we also detected a substantial amount of AAV backbone genome, so the viral vector that was used to deliver the CRISPR apparatus had integrated at the cut sites. And this would obviously be an undesirable outcome. And we confirmed that AAV vector integration by PCR also. So to kind of unify these concepts, I just want to draw your attention to the myonuclear domain hypothesis. And this is basically the idea that each myonucleus in a myofiber serves its surrounding area of cytoplasm in terms of the gene products that the muscle cell needs. And also, our data strongly suggests that dystrophin, mRNA, and protein aren't free to diffuse within the membrane or the cytoplasm of the myofiber. We observed that we have dystrophin expressing and non-dystrophin expressing nuclei coexisting in the same fiber. It's illustrating this cartoon diagram here, but also here's a single isolated myofiber that shows a very similar pattern. And what I'm now interested in is whether the spatial restriction of other gene products associated with these corrected and non-corrected and corrupted myonuclei. And if you imagine a situation where you have fibers where there's regions of protected sarcolemma next to unprotected regions, it seems fairly obvious to me that this would not be a desirable situation for a therapy, because the fiber is simply going to undergo damage at the unprotected region. And I think that this has really important implications for some kinds of CRISPR therapy, such as the double cut strategy, which I've outlined today, but also for cell therapy, where you introduce dystrophin expressing myonuclei into an otherwise non-dystrophin expressing muscle context. So just to summarize, exon skipping therapies are progressing in terms of the number of exons that we can target and the delivery technologies that we're using, with PPMO being perhaps the most advanced. Different patterns of membrane dystrophin localization are observed when comparing different kinds of dystrophin restoration therapies. And I think this is suggestive of an important factor, and potentially overlooked factor in the therapeutic success of dystrophin restoration therapies. And I think that these data strongly suggest that there's a major advantage of exon skipping over CRISPR-Cas9 therapies, at least the double cut strategy, which I've outlined here. And I will just acknowledge all of the people that were involved in the work and the funders, and happy to either take questions or pass on.
Hello, so I'm Linda, and I'm really pleased to be here, and thank you for the invitation to come and talk. So the title I was given is New Version of Exon Skipping ? So I wasn't exactly sure what I was supposed to cover, but I am going to cover where I think the challenges are for exon skipping and the improvements that are being attempted to try and address the problems. So these are what I consider to be the challenges. So it's the applicability to patients, so it's a mutation-specific therapy, as Tom's outlined. The efficacy is very limited, so again, as Tom's outlined, so it's good that he went first and explained everything. So the ones that have now been approved work in a very limited way. We're getting very low levels of dystrophin restored. So it definitely needs to be improved, the efficacy. And part of that is enhancing the delivery of the antisenses to the muscle, because basically antisense just produces some really expensive urine when it's given to patients. Most of it is excreted almost immediately. And it's not the best therapy for patients because it's a weekly treatment, because we're targeting the RNA. The RNA gets turned over, so the patients need a weekly infusion. So it's not the best for the patients. So steps to try and make the administration easier would be important. So they're my challenges. There's probably more. But for me, again, I've tried to think of how we can address these, and people are working on this, obviously. So the applicability, because it's mutation specific, there are other therapies being developed. some which could treat all patients, and we'll hear about them tomorrow, so the microdystrophins. And I will touch on in a second the scope for the ultra-rare exon skips. So as Tom suggested, there's a mutation hotspot in the dystrophin gene between exons 45 and 55, and many antisenses are being developed for those exons, but for exons in other positions on the dystrophin gene, there are not so many patients who are eligible. And then for the efficacy, improving the delivery, and Tom's already mentioned it, using antibodies and cell-penetrating peptides could really enhance that, and I will go through that briefly and then talk about using safe viral vectors for the delivery of the antisense, which shows some overlaps with using them for delivering the microdystrophin. I will talk about alternative chemistries and combined exon skips, and that's about it, but let me go through. So for the applicability, so as I said, there's a mutation hotspot, which if we could skip all those exons, we'd have antisenses, a collection of antisenses that would treat maybe 50% of patients. So, this has been worked out by Anna Mika, who will be here tomorrow. But if we could skip all the exons, individual exons that would restore the reading frame, we would have a therapy for 64% of patients. If we could do 2 skips, which won't be that efficient, but if we could do 2 skips, there'd be another 19% of patients who are treatable. So altogether, that would be 83% of all patients. But remember that it needs a new antisense for each exon we're targeting. So that needs to be optimized, put through clinical trial before any approvals. So here I've marked up the exon map for the rarer exons that have much lower applicability. So those circled in the blue would be applicable to 0.2% of patients. So I worked out on the incidence of Duchenne that would be about 730 patients worldwide. But at the bottom extreme, which is still very low obviously, but at the bottom extreme for the orange, for 0.02% of patients, that would just be 73 patients around worldwide. So is a drug company going to invest in the development of an antisense for such a small population of patients? And the answer is they will, but you have to be talking to the right company and the right research foundations. So I just wanted to tell you about, it was an antisense that was developed for a child who had an individual mutation. So it was an N=1, that's called. So this antisense was developed for this girl. So it's not for Duchenne. It was for a disease called Batten's disease, which results in lots of seizures. So you can see the timeline up there on the right. So she was diagnosed at the end of 2016. The mutation was identified a few months later, and her consultant thought, oh, this could be addressed with an exon skip. So he developed an antisense by the September of 2017, and then got approval after some tox studies to go into this girl, Mila, her name was, and a treatment for her. And it did work, it reduced the number of seizures. So there is situations where N=1 antisenses can be developed. So there is hope there. So moving away from the applicability, now looking at improving the efficacy. So there are 3 steps in my mind that we could use. So they're quite complicated terms, and I'll explain them. So stereo-pure antisenses, tricyclo-DNA antisenses, and mixed backbones. So I'll take you through each of those in turn. So the stereo-pure antisense, so these circles shown here, they're the bases that make up the sequence of the antisense. So they're DNA sequences, so they're individual parts of the DNA. And the way it's synthesized, normally you get a mixture of whether the bonds in between point up or point down. So they're called random. But this company, Wave, developed a synthetic strategy where they could have either all the bonds pointing up or all the bonds pointing down. So they're then called stereo-pure. And I'm standing in front of it. they developed an antisense for, again, not for Duchenne, it was for another target, and they compared it to the random conformation to ones where they're stereo pure, and they were able to show that their version worked better where it's stereo pure. So they've developed antisenses in a similar way for various exons on the dystrophin gene, and they did start a clinical trial for skipping of exon 51. This was, I think it was in 2019, so a few years ago, but within a few months they discontinued the trial because they weren't able to show any increase in dystrophin expression. So I think that's probably because exon 51, even with the random chemistry, it's a very poor skipper, and that's why we see very low levels in the epitalursen trials. But they have now, a year ago or so, announced that they're now developing a trial or entering a trial for skipping of exon 53. I wasn't able to find any updates on that, so I assume it's still happening and they're recruiting. And 53 skips better than 51, so it'll be interesting to see what they get out. So now switching to the tricyclo DNAs. So this is a completely different chemistry. So I should have said WAVE is using the PMOs. So tricyclo is a different chemistry. So don't worry too much about what it looks like, but it's just a different chemistry and it's cyclic. So Aurélie Goenval has developed this with Christian Laumann and tested it in the mouse model and is able to show good restoration of dystrophin, shown in the green, and was able to show that it targeted the brain dystrophin, so it seems to cross into the brain, which is important for cognitive defects. And they've now published another study, this was the end of last year, where they've conjugated, so they've bound the tricyclose to a fatty acid. And again, they show, it's a comparison between non-conjugated to conjugated, and they show a really nice increase, sort of a doubling in the amount of skipping in the MDX mouse. So it works well. And a company has arisen out of this work called Synthena. And in 2015, I have to say this was 2015, they announced they were possibly going into clinical trials. And again, I haven't been able to find any update on that. So I think they're still developing it, but I think tricyclose could be really important. And they have got, as a big consortium, a lot of investment, European money to look at the brain delivery for Duchenne. And then lastly on the chemistries is the mixed backbones. So again, don't worry too much about the structures, but in A, it's all the same all the way through. The length of the antisense. But in B, the ends are modified, the two bases on the end, the two circles on the end. And then in C, there's sort of, within the length of the antisense, they've introduced different chemistries. Don't worry too much, but it just means that it's not one chemistry all the way through. So here, they've developed this chimeric, so it's a mixture of what's called locked nucleic acids with 2'-methyl. So 2'-methyl was also put through clinical trial at the same time as the PMOs, so it's another good antisense, but not as yet approved. But they introduced these LNAs to enhance the stability and also the delivery in terms of how strongly the antisense binds to its target, which is really important. So it binds much more strongly, if that's English, stronger, and they give a higher percentage of dystrophin-positive fibres. So it works, it looks like it works and should be developed further. And then just beginning of this year, there's sort of a combination of the stereo pure and the mixed backbone. So this was a combined study between WAVE and Matthew Wood's Oxford lab, where they, again, there's some new chemistries down there in the corner, don't worry too much what they are, but they had various positions with that chemistry and then different stereo conformations along the length, and they did a massive comparison. And you can't see it at the bottom here, but they show a really good increase in the amounts of dystrophin exon skipping. So this is probably going to move forward, probably with WAVE. So there are steps being made to improve the efficacy of the skip. So now if we look at enhancing delivery, which obviously will enhance the efficacy as well. So Tom's already touched on the antibodies and the cell-penetrating peptides, but I'll quickly mention them as well. So as he said, there's Avidity and Dyne, who are using these antibodies to the transferrin receptor. So the transferrin receptor is highly expressed on skeletal muscle, and it does seem to be even more enhanced in dystrophic muscle, so it's a really good target. And they conjugate in slightly different ways, and they've got a slightly different antibody to the PMO that has been approved in the US. And this is the data from Avidity. So in the black is naked PMO. So this is in the MDX mouse. And in the red and purple is 2 peptide conjugated. So they give higher levels compared to the naked PMO. But with the antibody conjugated, this is the amount of PMO in the tissue, so it's how well they're delivered. You can see there's a big increase in the amount of PMO in the muscles, which is important. And then when they looked at the amount of exon skipping, again, it's really nice levels of restoration. We're getting 20 to 40% of dystrophin protein in the fibers, and there's a clear dose response. And the thing to note is that it was from a single injection, so 1 injection, and then they looked a month later. So, normally you'd have to do a weekly injection to see anything. So the PMO is getting in and staying and giving enhanced levels of dystrophin, which is really important. So just to give context, so Avidity also have a trial for another muscular dystrophy called myotonic dystrophy. So it's the same antibody delivering the antisense for the target there. And for any clinical trial, you'll hear about holds being put on clinical trials. So when they start a clinical trial, they will write the protocol to cover what they expect to see in terms of any adverse events to sort of deal with them. But if they see an unexpected adverse event, then the drug authorities will put a halt on the trial because they then have to modify their trial protocol to make sure it's safe. So when you see a hold being put on the trial, it's not always really bad news. It just means they're modifying it slightly. So they did have a halt placed, but it has now been lifted. So they are already in clinic for a different dystrophy, and they are now doing or planning the Exon 51 trial. So now looking at cell-penetrating peptides, which Tom has covered, but I just wanted to show you the Sarepta PPMO that they have. So again, in the MDX, they're showing really nice dystrophin staining in the fibers across a wide range of muscles, including the heart. And from that, they've gone into clinical trial. So again, they had, so this is for Exon 51, their PPMO for 51. They did have a halt placed on it, and that's because in patients they were or one patient, they saw this drop in magnesium in the blood. So they needed to modify their trial to look for that in future patients. And it is treatable, so they have a way of dealing with it. But it looks like it's working well apart from that potential side effect. So Tom's mentioned PepGen. They have also gone into clinical trials. So this is another peptide conjugated version for Exon 51. So they dosed in a control person and looked for safety, and it was well tolerated. And they're now in the first half of 2023 going to go into patients. So it's a really exciting time in a mutation-specific therapy. So lastly, for delivery, there's using AAV vectors. So as I said, these are safe DNA carriers. And in theory, because they transduce muscle very well, they target muscle very well, they will deliver to the muscles that need it and will give long-lasting expression. So it could, in theory, be a one-off treatment. So don't worry too much, but basically you package the antisense with a promoter to drive its expression into the AAVs, make the vectors, and then these are injected. So again, this was the work of Orally, and they showed that they did get good levels at 3 months of skipping, but by 1 year it was dropping off. So it looked like the AAV was actually being lost from the muscle, or they were losing expression. So it needed some optimization. So they have looked at that and now show long-term efficacy in a humanized model of Duchenne. So it's called the MDX52. So it's got a human gene. Actually, I'm lying. Sorry, take that back. It's not humanized. It is a mouse, but it has a deletion of exon 51. No, 52. Sorry, get there. Deletion of exon 52. And by skipping exon 51, you restore the reading frame. And they showed with their modification in the design that they got really high levels of skipping, so 100% in the heart, which is incredible. And it seems to be long-lived, up to 6 months. Obviously, we need to go longer term to make sure we're not losing the AAV, but it does look promising, and this was a fairly recent paper. So I know I'm rushing through a lot of stuff, so I do apologise, but you can ask me questions if you want more detail and things. So now I want to switch to the combination treatments, because it's not just the lack of dystrophin, it's what that lack of dystrophin causes to the muscles, so the wasting and the fibrosis. So we and others have looked at targeting what's called myostatin. So myostatin is a negative regulator of muscle growth, so it blocks muscle growth. So if we can knock myostatin down, we should remove that block, and then hopefully the muscles will be bigger and stronger. So down in the picture at the bottom, there are some animals, and actually a human who has a deletion in their myostatin gene, and you can see they're really muscly. It doesn't impact their health, they're just really strong. So that's what we want to do with our antisense. So for myostatin, it's a very, very small gene compared to dystrophin. And, you know, as Tom laid out, all the exons fit together. So you can see 1, 2, and 3 will normally fit together. But if we use an antisense to skip exon 2, then what's left won't fit together. So we stop the protein being expressed. So that's what we did in the MDX mouse. And we combined it with the myostatin antisense with the dystrophin antisense to restore the expression. So in the left-hand graph, we're looking at the force generated during contraction with repeat contractions. And you can see that the bottom lines are in the non-treated mouse, you know, they're the dark circles. And then the triangles, the empty triangles, are just the myostatin exon skip. So it doesn't work on its own. But when we combine the dystrophin with the myostatin in the dark squares, you can see we're improving almost back to what's seen in the wild-type mouse. So it works very well as a combination. And that's true also of the force that the muscles can generate. But, and Tom knows this, but targeting myostatin in patients actually isn't going to go anywhere, because we know that in patients, they seem to have a sort of compensatory mechanism for knocking down myostatin. So there isn't a lot of myostatin to target in patients. So that's not going to probably go near clinic. So we're also using antisense for targeting a protein or gene involved in fibrosis, which is another phenotype of the muscle. So here's a complicated pathway. Basically, it just shows you that TGF-beta, which was mentioned earlier, is the master regulator of fibrosis, and it feeds down through periostin to the fibrosis that's seen in muscle. Now, periostin, like lots of proteins involved in fibrosis, exists in different forms. So there's different lengths of the protein. And we know from other tissues that the protein that's been encoded by exon 17, shown in the blue in the middle there, are fibrotic forms of the protein, while those that lack 17 are non-fibrotic. So what we want to do with our antisense is switch the periostin from a fibrotic form to a non-fibrotic form. But first we wanted to establish that that's true in muscle, that the 17 containing is fibrotic and the non-17 containing isn't. So we wanted to show the levels of periostin in muscle in the MDX mouse compared to wild-type mouse at different ages, because obviously fibrosis increases with age. And you can see in that top graph there that 3, 5, 8 and 12 months, that the levels of total periostin is always higher in the MDX, which is shown in the red. And then when we look, which you might be able to see, but when we look at the exon 17 containing, again, there was much, much more in the MDX compared to wild type, and that exon 17 containing increased with age. So that was work done by a brilliant PhD student. She's just doing her corrections now. And another PhD student has developed this antisense to exon 17, and she's tested it in vivo in the MDX mouse. And so on the left is just the periostin antisense on its own. She's looking at the muscle activity in terms of strength and the ability, or not the ability, but how long the mice can run before they get tired. And the red bars are the things to look at compared to the blue. So the blue is treated with a scrambled antisense, so it's not targeting the periostin. And you can see that they're much weaker on the grip strength graph compared to the wild type. But with the periostin antisense, their strength is increasing and is fairly similar, not statistically significantly different to what's seen in wild type. And then again, for the distance they can run before they're tired, it's improved with the periostin antisense on the bottom there. And then in this graph, I know there's a lot of bars, but she was using the antisense to periostin with an AAV microdystrophin, which you'll hear about tomorrow. But basically, it restores dystrophin. She used 2 doses of the AAV, with and without the periostin antibody. And again, in the red bars, you can see that there's a significant increase in the grip strength and in the time to fatigue when there's periostin and dystrophin together. So it works well, and we're trying to develop that further, and she will now do a combined antisense for dystrophin as well as the periostin. So these are my future prospects. So Tom talked about CRISPR-Cas for gene editing. So CRISPR-Cas can be used in a number of ways to induce a permanent exon skip. So as Tom went through, you can cut either side to delete permanently that exon. You can also target within the exon to modify its sequence so it no longer looks like an exon and will get spliced out, which is sort of what we're doing with antisense. But cutting in an exon, because that's what normally codes for a protein, is risky. So definitely targeting either side of the exon is better. But I think we should, as a scientific community, start to look to use another Cas called Cas13. So the stuff with the DNA targeting is all Cas9. Now Cas13 targets RNA, so it's much, much safer. So you're not cutting the DNA, the coding sequence, you're doing the RNA, which gets turned over. Now there's what's called a dead Cas13, which is been mutated, so it no longer cuts the RNA. So we could use this to act a bit like an antisense to bind to the exon we want to skip, and it will mask it, and it will get skipped over. So I found this paper. It was a poster presented at a scientific conference last year, where they're, again, working on a different disease called Usher syndrome, where there's mutations in exon 13, which they block with this dead Cas13, and they show that in vivo it works very well. So I think that would be a really exciting new prospect for Duchenne. But again, it will be mutation specific and would need development. And that's me.
Thank you. Hello. Yeah, does anyone have any questions? Yeah, Darius.
Thank you. Tom, I have a question for you. Do you have evidence that your CRISPR-Cas worked on the myofibers rather than, for example, myoblasts? Because one of the possible explanations for the patchy appearance of dystrophin that you have was that you corrected in the myoblasts, which then fused with myofibers, and hence you have the patchy appearance. And the question related to both speakers is, what is really being targeted by these modified PPMOs? Is it any specific type of muscle, or is it sort of global? Because transferrin receptor is expressed on all, on myoblasts, on muscle fibers, and also on satellite cells. Actually, it's very important in satellite cells. It's critical for their survival. So, do we know anything about which cells are being targeted? Thank you.
Yeah, thanks. Great question. Obviously, this question of satellite cell targeting is a really key question for gene editing. The implication is that if you correct the stem cells in the muscle, these will continue to add dystrophin-expressing myonuclei to the fibers as the disease progresses. So this would be a beneficial situation. We did not test this. We didn't expect that it would work in satellite cells because it's been published that AAV is very poor transduction efficiency in satellite cells. However, one of the first papers from Amy Wage's lab showed that there was editing in the satellite cells. I think they saw, well, I don't think they used quite as quantitative methods as we have, but we didn't think the correction was sufficiently impressive to go and look in the satellite cells because we expected it to be a lot lower than what we saw in bulk muscle. But it's a key question. If those therapies are going to be developed further, obviously it would be great to get editing in the satellite cells. And it also speaks to, you put the paper up from Michael Rudnicki's lab as well about the inherent satellite cell dysfunction related to dystrophin. So obviously correcting it in the satellite cells may have that additional benefit there. Did you want to take the second question?
I haven't got anything to...
So it was about the type of...
Whether we know which cells are being targeted using those targeted approach. Because it sounds like muscle. But of course we know now that muscle is quite a complex structure in terms of types of cells.
Yeah, so I think that's we have a common opinion on that. we don't really know, sorry.
Does anyone else have any questions? No? Actually, I do, because again, being on the road, I've had quite a few questions about exon skipping, so I thought I could just bring some of them. And some of the kind of topics I wanted to bring on board, you've already kind of touched upon, but just for clarity, if you could just go over. I've been asked quite a few times with exon skipping why mutational hotspots are being targeted. And Linda, you touched upon eligibility, and you've talked about ultra-rare exon skipping. Is there anything more you'd like to say on, because I've met quite a few families with exons, of course, not in the hotspots. What do you have to say for that? Or?
So I think, yeah, if you haven't got a mutation that's applicable to a skip in the mutation hotspot, it's not good news. But I think from so I've recently been talking to a research foundation in the US who want to develop exon skipping for dystrophin exons that have really low applicability. So there are people out there who are interested in these ultra-rare mutations. So I've been working with a family who I actually met in an Action Duchenne conference a number of years ago to develop an antisense for exon 33 skipping. And, a gene edit as well for an exon 33. So I think, there are people who will look at those and try and develop them. And I know, and I hope I'm allowed to say this, but I think companies will engage with you on the rarer mutations for sure.
Actually, good. If anyone has a question, just shout it out. Otherwise, I'm going to kind of drive. Yeah, Darius, do you?
I think I'm very quickly turning into a hate figure in this gremium, but I have to ask these questions, I'm sorry. So with CRISPR-Cas approach, of course Cas9 is a bacterial protein. I've just mentioned that dystrophin itself can be immunogenic. I mean, it is immunogenic, right? The question is how quickly this response develops and how damaging it is. Now, Cas9 is definitely immunogenic, it's a bacterial protein. So by expressing in cells, it's very likely that they will be eliminated. So is there a risk that if you do Cas9 in satellite cells, you eliminate the pool of cells that are responsible for regeneration? So in a sense that you will actually make the damage worse and permanent rather than help.
Thanks for that generous question. Well, 100% agree with everything you said. Obviously, a large proportion of the population have already been exposed to Cas9 through, if you eat yogurt, this is a bacterial product, there's Cas9 in bacteria. So many people have pre-existing immunity towards Cas9-related antigens. So that means that the therapy is going to be very limited or impossible in those patients. That's a priori. So then the second thing is introducing foreign antigens, which then trigger a new immune response, which is your question. So would you get an anti-transgene response, which would diminish the therapy and, in the worst case, damage the satellite cell population? So I think the first thing definitely has been shown. Anti-transgene responses are known in the CRISPR field, and this is an issue that the field has to deal with. The second thing, whether it would, be harmful to the satellite cell populations, I've not heard that idea before. I think it's entirely possible. I think it's probably not the case because I think the delivery to satellite cells is so poor that I don't think, that's happening. I should say. I'm relatively skeptical of the CRISPR approaches in general, and I hope that was sort of the message of my talk. I think CRISPR is a huge improvement and a tool that we can use for research purposes, and it will change medicine in some ways, but it's not a magic bullet that solves DMD. I think it's really important that people understand that. And I think that anti-transgene responses, pre-existing immunity are major hurdles to the successful translation of the therapy. So I completely agree there. We have not looked at any anti-transgene response, so I can't comment on it in our animals, but yeah, hopefully that answers your question.
Tolerance injection and tolerance induction. Well, that's why I mentioned tolerance induction earlier and probably introducing the treatment early on, because then there is a chance to tolerize the patient either or both to the CRISPR-Cas delivery tools as well as dystrophin. So I think that's one possible explanation. But there maybe I'm looking forward to the next sessions because I'm hoping that there will be some very clever approaches that are being developed to solve this problem. Thank you.
I just have one last comment on that. Sorry. That's indeed what you said. Treating early to tolerize has been demonstrated in animals, I think by Eric Olson's lab. So that's absolutely 100% true. Obviously, with our mice models, we can inject them on day 2 or in a push day 1 of their lives. That's obviously not possible in a DMD patient where you have to wait for a diagnosis, which is going to come later, unless we have newborn screening. Yes, which obviously is a huge. So you're preaching to the converted with me. I believe it also. This would be a game changer potentially if we're going to progress these kinds of therapies.
Okay. So I just. Oh yeah.
Sorry, can I ask a question?
Of course.
It was regarding this dialogue. It's very interesting that you're talking about here, but there's some terminology that I don't quite understand.
Sure.
So. I don't understand the concept of satellite cells. What do you mean by that? I understand stem cells, certainly.
So satellite cells are the muscle stem cells. Right. So they're called satellite because they're kind of, if you imagine, sort of on the outside of the fibers. So you've got sort of tubular fibers, and then there are resident muscle stem cells sort of peppered around these fibers, and we call them satellite cells. But it basically means muscle stem cell.
They're still contained within the muscle.
They're still in. Yes, exactly. If there are any technical questions like this, very, very happy to take them either now or immediately after the session. Please, I'm sure any of the scientists would be happy to.
No, so I was just asking the question about the mutational hotspots being targeted and the rare exons not being targeted. But I wanted to also ask that I've met quite a few families and individuals who've got multiple exons missing or mutated. Do you have anything to say about multiple exon skipping therapies? Is that something?
So, yeah, the mutation hotspot, the 45 to 55, people are targeting all those exons in the hotspot. So it could be a therapy that's straight off the shelf, applicable to, 65% of patients. But unfortunately, with each exon you're trying to skip, the efficiency goes down because remember, you want to target the same piece of RNA. There's no point doing one exon on one piece and another exon on another. You're not going to restore the reading frame. So where you need a multiple exon skip, the efficiency is just really low. So that needs to be optimized and developed a strategy to try and improve that. And I know there have been a number of papers looking at that multiple exon skip, but it will be a while before that's near clinic, I would say.
Absolutely, thank you. Linda, you did say that exon 51 is a poor skipper, and you said 53 skips better than 51. Sorry, could you just elaborate?
So I think 53, and this is just sort of generalizing a little bit, but 53 seems to give twice as much dystrophin protein compared to 51 in the trials and in the approvals that happened. So it was 1% or less than 1% for 51, and then 2%-ish for exon 53, which is still really, really low, but on the basis that 51 had been approved, 53 got the approval. And the reason that 51 doesn't skip as well as 53 is down to the sequence of the exon. So the antisense needs to bind the exon it's targeting, and that's the strength of binding is down to its sequence. So in DNA, there are 4 letters, C, G, A, T. So C and Gs, they will bind very well to their partner, as it were, so you get much stronger binding. So it looks like there's probably more C and Gs in exon 53 than there is in 51, and probably the folding of the RNA is different, so the target's not as accessible. So it's a combination of things, but it's probably down to the sequence.
Thank you, that really clarifies a lot of my kind of question. I just have one last question, unless someone else has any. Okay. This is, again, a general question as well. We see with different therapies, certain age groups are targeted, or whether ambulatory, non-ambulatory. With exon skipping, how much impact does the health of the muscle have? So it's just, could you just elaborate more on that? And do we want to target earlier on when the muscle is healthy? So we do see that dystrophin restored, and that's where it's going to sit. So if you could just talk a bit more about that, because that's a concern as well. Yeah.
So, I mean, I'll give my opinion on that. I mean, I think that muscle quality is a key factor in the success of exon skipping. So, over time, the quality of muscle in patients is declining, and that's as a consequence of adipose deposition, so what we call fibrofatty degeneration. So there's an increase in connective tissue in the muscle, and there's an increase in fat in the muscle. So this is basically decreasing the number of myofibers that you have in which you can do exon skipping. So there is a bit of a debate about this, but I mean, treating earlier where there's more muscle, you're going to get more dystrophin. But later in life, there are fewer fibers in which to do the skipping. So it seems to be that there's an obvious benefit to initiating treatment earlier, if possible. Not to say there couldn't be a benefit from treating in the later stage as well. But
yeah, I mean, that's basically what I was going to say as well. But as I touched on at the end, maybe combining the dystrophin skip with something that's targeting the fibrosis pathway or the wasting pathway could be really important in enhancing the dystrophin.
Is that what you mean? Something like steroids?
Well, people have to be on steroids, don't they, before they're treatable with the exon skip. But I think it's, targeting the components of the fibrosis pathway to stop the fibrosis and just keep the muscle in a better condition. So, you know, combination treatment might work better than the dystrophin antisense on its own.
Thank you very much. If no one has any other questions, we'll close this session. Thank you both so much. That was really informative. And we can break for tea. Thank you very much, everyone.